Good morning, college biology students. I'm going to dive into a discussion of the membrane and how things move through the cell membrane. So this is chapter 5. So the first part, we're going to describe the components of biological membranes, including the cell membrane, so what it's made out of, and then kind of relate it to the fluid mosaic model, which is just kind of an analogy that we often use to explain that structure of the cell membrane. So you will notice, so this is just a little cross-section piece of the cell membrane, and it shows how complicated that structure is and how many things there actually is embedded and attached to and so forth of that cell membrane. So if this is that phospholipid bilayer, which we're going to talk about, you can see that there's things called glycoproteins, and then there's globular proteins, and there's protein channels, which like a tunnel going through that cell membrane. Integral proteins, peripheral proteins, so peripheral means just on one side. Glycoproteins, cholesterol, which, I mean, cholesterol gets a bad rap, but we actually need cholesterol in our cell membranes. There's carbohydrate markers here, and so on and so forth. So just really a lot of stuff that we're going to talk about in here, and the importance of those different components as well. So the structure of the membrane, we do use that term fluid mosaic model. Oh, my face is right in the way. The fluid mosaic model, if you think of a mosaic in art, so Ava, I expect you to be really knowledgeable about that. So the fluid mosaic model, you know, a mosaic where you cut pieces of glass and you kind of can piece them together to make a pattern or images and so forth. So that's what we think of the cell membrane. There's just a lot of pieces that are important and have really important jobs within that cell membrane. Obviously, a phospholipid bilayer. So each of these little components, I'm going to go back because it's more prevalent on here. So we do say it's made of this phospholipid bilayer. So we see these hydrophilic heads versus hydrophobic tails. So the hydrophilic heads align so that they are facing the intracellular and extracellular fluids, which are high in water. So these hydrophilic heads are water loving, and we see them on this side, and then we see them on this side. It's a bilayer, meaning there's two layers. The hydrophilic or water fearing, oh, sorry, I think I said that wrong. Sorry. Oh no, I said that hydrophobic tails are to the inside. They don't like water, so they're always going to align so that they're pointed to the inside. I just like that visual. So that phospholipid bilayer of those molecules exclude water-soluble molecules from nonpolar interior of the bilayer and the cell. New research and this is new just within the last decade, suggests that parts of that membrane have little microdomains that have like specific composition. So maybe it's like 2 particular proteins and 2 or 3 particular lipids that all seem to be grouped together. So we call that a domain. We did talk about domains in terms of when we talked about proteins, about repeating domains and common domains and so forth. But yeah, we think even though this cell membrane is fluid, so most of these proteins float around and they can move all over the place. All of these things, in fact, in the cell membrane seem to float around, and because this is liquid, it's like when you put oil in water, it's a phospholipid. But these microdomains, there might be, say, like this little grouping here might just kind of always sort of, this one, and this one might have a little connection to each other where they somehow stay in that domain together. And that might be important to the cell membrane function, depending upon what the jobs are of those particular embedded molecules and structures. Transmembrane proteins transport molecules either actively or passively through the membrane. So examples of this are like sugar transport, like glucose. Sodium and potassium channels in the nerves become very important. And then heart and muscle cells. I gotta move my screen a little bit here. So these Sodium potassium channels in nerve cells. This is really important. If you're an athlete, you worry about keeping your electrolytes high enough for your nerves to function properly. Sodium and potassium are some of those electrolytes. So if you drink Gatorade or sports drinks and they taste a little bit salty, it's because, yeah, they have sodium and potassium, and that's kind of that flavor that you're tasting. But those are really important in the function of nerve cells. If your sodium and potassium levels get too low, if you've ever seen someone get badly dehydrated, they actually lose muscle function. It becomes difficult to control muscle movements, and it's obviously really a problem. So those are very important, that sodium and potassium pump, if you will. Interior protein network determines the shape of the cell, and it also anchors or keeps certain proteins in certain parts of that cell membrane. So I said when some of these, a lot of these things in the cell membrane can sort of float around and move around, but some of them are anchored in place and they can't. So like the shape of red blood cells, which I actually have over here, I always say they look like a donut, but the hole doesn't go all the way through, and that's because the nucleus dies off in them. But that shape is pretty important for carrying oxygen and also nutrition through your body. So that shape of red blood cell is important, and there's interior protein network that helps it keep that shape. Get my face out of here. And then cell surface markers, and there's lots and lots of cell surface markers. The most common ones I think we think of are these ABO blood group markers that determine blood type. I'm not going to spend a lot of time talking about that, I don't think. But I do want to just show you really quickly. I like this visual because it gives you kind of and again, I'm just in the way here. All right. Red blood type cells are your blood type is either type A, type B, type AB, or type O, and then there's a positive and negative Rh factor also associated with that, but we're going to ignore that for right now. But if you are group A, if you have blood type A, you have a particular surface marker, and then you have antibodies that will attack type B markers. So you can see these antibodies. So you have antibodies in your blood that recognize type B blood as foreign, and you can see, because these are kind of diamond shaped, and then these have the edge of that diamond shape. So the antibodies are this shape. The antigen, which are attached to your red blood cells, are this shape. Group B is the opposite. They have antigens on the cells. That's their own cell surface marker that are diamond shaped, but then the antibodies attack the shape that is on the group A. So if you're type B blood and you somehow got a transfusion of type A blood, it would cause a lot of problems in your body because it would attack this blood, recognize it as foreign, and that actually causes problems with your blood. It actually causes some clumping and clotting that actually would kill you. So you don't want to get the wrong kind of blood. If you are type AB blood, you have both of those types of antigens, but then you don't have any antibodies. So you can't, obviously you couldn't have anti-A or anti-B antibodies because you would be attacking your own cells, and that'd be a problem. And right here is why group O blood are universal donors. Okay, so if you're O, and specifically if you're O negative, so I'm O positive, so I'm not quite a universal donor, but my daughter is O negative, so she is. She's a type O, so her and my blood cells do not have any surface antigens on for identity. That's why you can give type O blood to any of these other blood types, and it's not going to react because this actually has no surface markers at all. So that's just a quick little overview lesson of the different blood types. This is mentioned, I think, in your book as well. Electron microscopes, which are very expensive and very large, actually have provided some structural evidence of cell membrane structure. We can't see individual atoms, of course, really with it, or at least not that structure, but we can see a lot of the parts of the cell membrane. So this was really integral in kind of proving and discovering more about that cell membrane. So transmission electron microscope, there's two different types of electron microscope, either a transmission type or a scanning type. Transmission type is a specimen is sliced into very thin slices, and then a beam of electrons is directed through the sample. And we can see if you kind of recompile those slices, if you will. A scanning electron microscope is looking at a very tiny organism or object in three-dimensional, and the outside surface of that is actually sprayed with metallic dust, and then the beam of electrons basically scans that outer surface to give you a three-dimensional image. So that's how they're different. All right, section 2 then, phospholipids, the membrane's foundation. So we're going to talk about, we're going to list the different components of phospholipids, explain how membranes form spontaneously, and describe the factors involved in membrane fluidity and kind of how to maintain that, or how you might upset the integrity of the membrane as well. All right, so phospholipids are obviously the membrane's foundation, like that's what it's made out of. And I already said we have these, so they're composed of, they're a bilayer, so 2 layers of phospholipids, a polar hydrophilic head and non-polar hydrophobic tails. And just kind of what that structure looks like. This is kind of what that molecule actually looks like. And so membrane fluidity can sometimes change a little bit, especially if exposed to certain environments. So a saturated fats tend to make membrane less fluid because they pack together well. So I added a little bit here. The cell membrane lipids are synthesized in the endoplasmic reticulum. I think that's important to mention. And then they contain both saturated and unsaturated phospholipids. If you remember the difference between saturated and unsaturated, and then we actually have a lipid identifier. All right, so I don't want to draw all the rest of it because it gets like much more lengthy than this. But a hydrocarbon, so this is a saturated lipid because all the carbons are single bonded, and then there's the maximum amount of hydrogen saturation that there could possibly be. And these are very straight molecules. If we have an unsaturated carbon, which would mean maybe there's a double bonded carbon right here, and then we don't have quite as much oxygen on here. This molecule is more kinked. It actually will kink here instead of being in a nice straight line. I have too many bonds on my carbon. If you remember, carbon has 4 bonds, so let's take one of those off too. But anyway, so this would be a kink in the molecule right here, and this would make our membrane a little bit less fluid. So that's the difference between, a saturated and unsaturated. So these unsaturated hydrocarbons are actually pretty important in the cell membrane and help maintaining its fluidity, if you will. Because a saturated fat tends to make membranes less fluid because they pack together so well. And then changes in environment, changes in temperature or pH have a really big effect on that lipid. just think in general how fats are affected by change in temperature for sure.